Right, so hello everyone and welcome to today's MASTS webinar. My name is Phil Bell-Young and I am going to be today's host. Our speaker is Chloe Cargill from the University of Aberdeen, who will be speaking to you today about a collaboration towards understanding movements of black-legged kittiwakes across the North Sea. But before we get started, we're just going to go over a few quick housekeeping notes. So, after Chloe's presentation, there will be the opportunity for a Q&A session. So if you have any questions at any point, if you'd like to write them in the Q&A box that you should hopefully see within your Zoom box, well, you'll see that at the bottom of the screen. You can submit questions at any point throughout the talk, and we'll come back to them towards the end. So this session is scheduled to end at around 1:30 p.m. UK time, but depending how many questions we get, we may run over. What I will say is that the webinar is being recorded. So if you have to leave early or you missed something or you want to share this with a colleague who wasn't able to join today, you will be able to find the recording on the MASTS YouTube, as well as all the previous MASTS webinars on our YouTube as well. So with all that said, I won't kitty waiting any longer. So a quick introduction about our speaker. Chloe is I'm going to say this word wrong. Chloe is an ornithologist, primarily concerned with the conservation of coastal and pelagic taxa. She is currently a second-year superdoctoral training partnership postgraduate researcher focusing on enhancing black-legged kittiwakes meta-population modeling in the context of global change and renewable energy. Chloe is also part of a working group dedicated to establishing the status and viability of the remaining breeding colonies of the Hutton shearwater in the Kawakora seaweed ranges in New Zealand, and has previously worked on the distribution and breeding success of the little penguin in the same area. She holds an honours degree in conservation biology and ecology from the University of Exeter, and she has a master's degree in marine ecosystem management from the University of St Andrews. And with that all said, please give a massive virtual welcome to today's speaker, Chloe Cargill.
Awesome, thank you. All right, yeah, hi everyone. Thanks for joining today. So yeah, my name is Chloe, and I'm a second-year PhD student at the University of Aberdeen, and currently also a visiting researcher at the Scottish Oceans Institute at the University of St Andrews. I'm really grateful to be here with you today to share some of this exciting work. So the black-legged kittiwake, it's a seabird, a medium-sized gull, which is widespread in the Northern Hemisphere. It is classified as vulnerable by the IUCN, and it is red-listed in the UK as a bird of particular conservation concern, primarily because there are populations in decline across its breeding range. So the overall aim of my PhD is to improve our current understanding of how populations of kittiwakes are connected, and we are particularly interested in whether genetic data could be a valuable resource for generating estimates of connectivity. For instance, can we use estimates of immigration and emigration derived from genotype data to parameterize a metapopulation model? Further, does this reduce uncertainty in subsequent estimates of population trends? I should state for clarity that we're not looking at temporary migratory movements, for example, the annual seasonal flow of kittiwakes between coastal and offshore regions. Here we are specifically interested in estimating the fraction of breeding adults in, for example, population A, which are derived from population B, and vice versa. So breeding movements between kittiwake populations occur most commonly at local scales. Apologies, okay, not sure what happened. So yeah, breeding movements, they occur quite commonly at local scales, but anecdotal evidence from color ringing and tracking studies suggests that the kittiwake has the capacity to disperse across larger spatial scales, such as between the Norwegian and Scottish coastlines, at a higher rate than we previously thought. So our question is, and this is particularly pertinent to the present day, given the expansion of offshore wind developments across the North Sea and the policy underpinning the licensing and the mitigation measures of these installations, should population assessments for this species be looking at taking into consideration the fate of populations elsewhere, even those located across legislative boundaries, and at what spatial and temporal scale is it appropriate to do so? So to begin answering this question, I was very grateful last summer to receive funding from the MASTS-SFC Funding Council Saltire Emerging Research Scheme to set up a collaboration between the University of Aberdeen and the Norwegian Institute for Nature Research. I would like to open this presentation by giving a really big shout out to my supervisory team at the University of Aberdeen, the University of the Highlands and Islands, Marine Scotland Science, and the Joint Nature Conservation Committee, the collective funders of my PhD, and a really huge shout out to Signe Christensen Darsgaard at the Norwegian Institute for Nature Research for hosting me in Norway last summer, and as we will see later in this talk, for continuing this partnership beyond the scope of the award itself. Okay, so a bit of need to know background information on the kittiwake. So kittiwakes take about 3 or 4 years to reach reproductive maturity. Prior to this stage, as juveniles and subadults, they will learn skills necessary to survive and gain a mate, develop adult plumage, and decide where to make a first breeding attempt. So on your screens, there is a simplified visual representation of a single population. The solid black circle around that population indicates a closed population with no immigration or emigration of individuals, where juveniles are aged 0, immatures at 1 year, subadults aged 2 years, breeding adults at age 3. So now think of time moving forward, and at each time step, so here that is 1 year, the number of birds in each age class and overall the total population size is determined by survival rates at specific age classes and the productivity of the adults. So we can apply this structured representation of the population and how it changes over time to explore drivers of historic population trends and to predict the fate of populations into the future. At present, population assessments used within policy treat populations as closed units. For example, when estimating the effect of different levels of anthropogenic mortality or environmental change on the species. So this is a problem because it potentially leaves out an important aspect of realism in this study system. We know from color ringing and tracking studies that kittiwakes move between populations, leaving us uncertain as to whether we are taking the most appropriate measures for the species. However, it remains a challenge to include measures of connectivity between populations because it is quite hard to appropriately observe or capture such movements using color ringing studies and GPS data. So the purpose of this Saltire research exchange was to collect genetic samples from kittiwakes breeding in Norway to complement samples that have already been collected from kittiwakes breeding in the UK. We are interested in evaluating the presence or absence and the magnitude of population connectivity over that large spatial scale. In summer last year, I joined a field team from the Norwegian Institute for Nature Research to collect samples from urban kittiwake colonies at two locations in Norway. So that was Råvik and Ailsund. And I kind of gave you the approximate locations of these colonies in the map on the right-hand corner of your screen there. So we collected feather samples from the colony environment and from individual birds whilst they were being handled for ringing. Our present focus is population level data. However, a kind of downstream benefit of associating genetic data with individual coloring codes at this stage is the potential for looking at patterns of connectivity between populations in association with individual histories of breeding effort, breeding success, and survival. So last summer's visit was a really great starting point from which to grow this partnership between the University of Aberdeen and the Norwegian Institute for Nature Research. So for instance, if I could once again draw your attention to that graphic in the lower right-hand corner of your screen, ongoing work now includes the collection of samples from further locations across the Norwegian coastline, and around the UK, in addition to the southern North Sea, 1 or 2 offshore sites, and actually a bit further afield into the West and North Atlantic, which are not shown on the map here. So this will allow us to understand better the regional and large-scale dynamics for the Atlantic subspecies of kittiwake as a conservation unit. So we are particularly interested in the implications of this work for providing insights into how population level effects play out at the level of the metapopulation, and by which I mean a collection of interlinked populations. We can assume that metapopulation models of this long-lived bird are likely sensitive to immigration and emigration of breeding adults, because closed population models are highly sensitive to adult survival. And what we are really interested in is whether real values of immigration and emigration are such that, to give an example, a change in population parameters, such as breeding success at colony A, has a knock-on effect to the fate of colony B. Or do immigration and emigration between kittiwake populations operate within a safe space, whereby it is what is happening at the local level which is most important to the fate of the colony, and therefore most important to the species conservation and management. So we really hope that the implications of this work within policy could be really interesting. So for example, better understanding of contemporary extinction colonization dynamics is really highly relevant to decisions around the decommissioning of offshore structures. So you see here, these are kittiwakes nesting on oil rig platforms over the Norwegian shelf. There is a huge potential to fully collaborate with industry professionals and to work together to understand the status of recently established offshore colonies. For instance, are these aggregates, do they function as an extension of a single local colony, or do these offshore structures support birds, for example, Shetland and Norway together? Now, an additional important question to ask is, are the current and static protected areas networks appropriate for species conservation? Once one starts to consider the importance of the influx of birds from further afield, not to mention the potential for population connectivity to be dynamic over space and time. Further, there are really clear implications of this research within the arena of marine spatial planning and licensing for offshore wind developments. For example, in the future, we hope that studies such as this will aid developers in ensuring that they are being the most effective with regards to the placement and extent of ornithological mitigation measures. So, for example, a recent study by Hugo Putahina, David White, Susan Govanek, and Fraser Stewart at the University of Southampton was recently published in Renewable and Sustainable Energy Reviews, and that illustrates that the available space for the required offshore wind developments to meet net zero targets is already highly constrained by the existing pressures of human activities on the North Sea. So what I really hope is that there is a time in the not too distant future where the data outputs from my PhD and future work such as this will combine with advances in ecosystem-based cumulative effects modeling to identify the appropriate placement of developments that will enable net zero targets to be met alongside biodiversity conservation targets. So this is just a snapshot of the work that I'm getting on in my everyday at the moment. And I'm really looking forward to interpreting these findings in the context of the populations that these data represent. So for example, looking at the history of colony counts and breeding success over the time period around which they were sampled. But I'm going to round up with what work is currently ongoing. I have 3 work streams sort of running alongside each other at the moment. Firstly, developing an appropriate workflow for integrating genetically derived estimates of connectivity and the uncertainty around them into a metapopulation model framework. Secondly, I'm using a simulation-based approach, using these models to explore further what kind of spatial scales are appropriate to think about in the context of population assessments, especially given the expansion of offshore wind in the North Sea and other shelf seas across the North Atlantic Ocean. And thirdly, we are starting to receive new samples from within the UK and further afield, which I'm going to be inventorying and processing in the lab at the end of this year and spring next year. And I just wanted to highlight during this talk that once I finish with those samples, so once I've extracted the DNA that I needed, the rest of the samples, so the feathers themselves, will be held in storage with metadata. And I'm really hoping that the aim is to put this inventory of samples online and to kind of put out a call for people interested in using these feathers for a large scale spatial isotope analysis. So I'll just highlight that to you there and say do follow updates on this for access to that if you're interested. So I'd like to give a huge shout out to the network of really amazing people around these shores who are out doing their fieldwork this summer. I understand that there's starting to be some issues around avian flu, which unfortunately is not unexpected. But hopefully they're out there gathering samples from kittiwakes. And this sampling effort has really been made possible by funding from the British Trust for Ornithology and the Seabed Group, and is really due to the enthusiasm and interest of over 60 like-minded ornithologists and researchers out there. So we have been really lucky in that we are able to take the project aims of this Saltire exchange funding program further in both time and space. And it will be really, really exciting to compare patterns of population connectivity at 2 distinct time points. So thank you everyone for coming along today, particularly those of you who are using up some of your lunch hour. This has been a really great opportunity to share what is going on. I think it's really important to build into future policy the relationships between populations and these dynamics over time and space. I'm really excited to be part of this next step towards this for the black-legged kittiwake, and I'd like to end with a final shout out to the network of people and organizations who are supporting this work. So if you're finding this interesting, please do follow the project on Twitter. The tag is at Meta Kittiwake. And please feel free to reach out by email if you'd like to chat or ask questions, or if you have some ideas that you think could be useful for integration into this project. My email and the Twitter has been on the slides throughout. So, okay, I'm looking forward to hearing your thoughts and hopefully to answer some questions, which now I can see a clock, I think we'll have more than enough time for. So thank you so much. Cheers.
Thank you very much. Wonderful talk. I'm sort of still, I'm brand new to MASTS, so I've not yet had an opportunity to actually see all the different types of research going on. I know the different areas that we do research in, so to see something like this is absolutely incredible. We've had a couple of questions come in, but I'm going to give people just a little bit more time. Again, if you do have a question, it's the Q&A box at the bottom of your screen. Now, I have a question to sort of kick things off, and I think actually you touched briefly at the end there about a potential answer to this question, which is all to do with the genetic data and the actual samples that you're collecting. So what kind of samples are you using to generate this genetic data that you're collecting?
Sure. No, thanks, Phil. Yeah, so I mentioned briefly that we're mainly looking at feather samples, and this is what I mean by feather samples is literally a feather from a bird. Sometimes this is collected opportunistically during ringing. Sometimes we're collecting it from the nest of the bird, sometimes from the colony environment. And there's like a number of reasons behind this, but it's on purpose, as in we are quite interested in seeing whether we can do this kind of genetic analysis using fairly low quality samples, for example, not as invasive or as stressful for the bird as taking blood samples. But there have been a couple of interesting publications which have given some like user-developed frameworks for extracting high quality DNA from the very tiny residual bit of skin and blood cells that's within the shaft of a feather. So yeah, I mentioned this for a couple of reasons. One, we would like to promote if possible the use of less invasive sampling techniques. Two, because of that, we can sample a lot more birds and sample more widely across the range. There's less, concerns in terms of like sample storage and processing, and you get the rest of the feather left, which is great, which means we can store that and look at stable isotope analysis in the future, or someone else can.
Well, we've got some questions coming in, and keeping an eye on that time, I'd like to kickstart. So we've had our first question, which hopefully should appear if I click this. So does the number of adults times productivity equal number of juveniles, in parentheses, the first population slide? Surely number of juveniles is equal to the AON's time productivity. Number of adults at a colony isn't necessarily a measure of AONs or of recruitment of juveniles into the population. So that's quite a big question.
No, that's cool. No, that's cool. I understood that. Yeah. No. The asker of that question is absolutely right. I was just didn't want to go too much into detail, so I just thought I'll go number of adults. That'll be enough for the audience. But you're absolutely right. I think it is common practice to use AON, apparently occupied nest, as a proxy for your sort of breeding effort. And yeah, you're correct. So that breeding. The breeding pairs times the breeding success should give you number of juveniles to add to your population at the next time step. Yeah.
Brilliant. So next question, which comes with a thank you for the presentation. Sounds like a really interesting project. So that's always nice to hear as well. Do you expect a change? Do you expect a change in the population connectivity graph that you showed between 1999 and 2001 samples versus the one that you are collating now? For example, more connections because of more offshore structures.
Yeah, it's interesting. I think you can go a few different ways with coming up with hypotheses for dynamics and population connectivity over time. I think there will be a change because I think it's likely that the movements between populations are really sensitive to what's going on at those particular populations at the same time, if that makes sense. So, for example, I was looking at a different, like a more regional analysis of movements within Norway recently, and I could see that there was this real big movement of individuals from one particular population in the coastline that was spreading out to the others, and I was, oh, okay, interesting, not sure about that. But then when I chatted to my collaborator about it, they noted that at the time of sampling, there was a particular piece of policy which had been changed with regards to predation, with regards to conservation of sea eagles, and that the predation pressure at that colony was quite high and breeding success for the kittiwake was quite low. So when you start to link it back to potential drivers of dispersal for kittiwake, i.e. lower breeding success or breeding failure, those drivers will, I think, be quite dynamic over time. So I think, yeah, outwards movements will be dynamic. And I imagine that where the birds choose to settle as well, what denotes a good quality colony, will be quite dynamic too. So I think it's going to be one of those things where you really have to be careful not to over-infer what's going on. But I think overall, it's going to be really interesting to get the first picture of what the movements look like.
Okay. So moving on, keeping my eye on that time still, but moving on, we've got another question. So what are the consequences, let me make that live, what are the consequences for your study of not obtaining genetic samples from all possible connected colonies?
Sure, yeah. Well, I mean, yeah, so you're right, we cannot, okay, so when you're looking at doing it, you want to understand where birds are going to different places. There's always the likelihood that you won't be able to sample at the colonies that they're actually going to, because you don't know where they're going to, right? So what we will do is we will try and do our best to represent the unit for the North Atlantic kittiwake subspecies, and we will do our best to get a representative sample of the sort of more the national areas that we are interested in. So we've managed to get quite a good scatter across the UK and along the Norwegian coastline, and hopefully this will just give us, it will give us a really good idea of the general direction, the flow and magnitude of where birds are going. Yeah, there will be some populations we don't sample, and there will be, it might mean that we kind of miss yeah, we miss some understanding of where they're going, but I think overall it's impossible to really track all of them, and I think the outputs will be interesting regardless. I think the main problem is if avian flu really kicks off and we can't get samples from a lot of locations, and therefore it will really limit where we are, limit within the connectivity analysis the populations we can include, in which case we might really be missing some big, like the key areas. So basically it will just limit our inference in different ways depending on where we get sampled. That was a bit of a messy answer, but I hope I am thinking about it, which is
Yeah, I mean, it's a complicated thing because, as you say, you can't always know where every single bird goes. So yeah, it's a very complicated answer to a complicated question. We do have more questions coming in. Just to remind people that we are recording this, so if you do have to go, because it is officially past, if you do have to go, that's fine. You can check the recording later. So our next question. I also wondered if you use tracking/tagging data to measure connectivity. So yeah, an alternative approach to it.
Yeah. So the reason for looking at genetics is because there have been a good couple of studies with regards to the color ringing to suggest that the amount of time and resources that are put into that makes it quite unfeasible to really capture, capture where the birds are going. A bit similar to what I just said with the genetics, I guess. But with the tagging, the GPS tagging, the studies that are going on at the minute, that what you can do with the tags is fairly small scale in terms of like putting on the birds within the breeding season. The equipment kind of expensive. And there are stress considerations on the birds to take into account. I think what one could do is definitely collect and try and integrate long-term color ringing data sets that we do have. If people are keen to do that within a network to I would say to inform what we're looking at with the genetics as well. I think this should kind of be done together, purely because of the gaps in what you can infer using each method. I think each method has its place, and the strongest way of using them all is to look at them, infer from one method in the context of the others as well. Yeah.
Yeah, it's an interesting idea because that would make lots of sense to have taggings, but yeah, there is that kind of, if you interrupt with the bird itself, are you actually affecting the data you collect, which is always something that's interested me actually. So next question, we have kittiwake populations trends have varied over time. Do you expect genetic exchange to vary with a population trend?
Sure. Yeah, I guess this links back to the would you expect to see a change in connectivity over time? Yeah, I mean, okay, so yeah, I would expect maybe from a declining. So yeah, if you have colony A, which is declining, and colony B, which is increasing, then I think there's a number of things that could, you might want to see from that genetically. If B is declining, are birds taking off and going somewhere else? If A is increasing, are birds coming in from somewhere else? Where, but where are those increases coming from? Is it lower breeding success that's causing the decline? Overall, I think if a colony is in bad health, there's likely to be emigration of birds from that area to higher quality breeding grounds. So this is this is why I definitely want to look at these connectivity maps in the context of the breeding success and colony counts, because I think you can only get a clear picture of what's going on looking at what could be driving dispersal and whether there are any density dependent effects to take into account at the source or receiving colony as well.
Brilliant. Moving swiftly on, because the questions are still coming in. There's some great questions here as well. This one starts off with, Great presentation, Chloe. Again, always nice to hear. I have another question. If there's time, yes, there is. Do you expect to see more migration following the impact of HPAI?
I mean, I think, yeah, so more long-term breeding movements overall. I think, yes, I think purely from my own reading and kind of background knowledge is that if HPAI is affecting breeding success at a colony is quite, it's public knowledge of prospecting birds will, try and use that what's going on at that colony as an index of whether it's a good place to settle down. And if you're getting lots of failed nests or chicks, high chick mortality, then yeah, I think birds will potentially look and take off, find somewhere else to breed. Whether they'll then come back, they'll probably come back in the fullness of time, probably. But yeah, which is why it's really cool in a way to be sampling this summer, which is kind of like morbid but interesting. Yeah.
Fantastic. So I think that is our last question. We have had other people write some comments. There's some very lovely comments, which I'm sure you'll take some time to read at the end. But basically people just saying that it's a super talk, really interesting, and they've had a great time, which I would like to emulate. Thank you so much for doing your talk today. It's been really interesting, once again, just to hear the kind of work that's going on here, and especially work that is not just, it feels very kind of multidisciplinary. It feels very much like it's not just a one area sort of form of research, and there's many connections. So I've learned a lot today, and I'm looking forward to learn a lot more. So I'm going to end our webinar there. So I'd like to start by saying a massive thank you to Chloe Cargill once again for joining us and presenting her work. I'd like to thank everyone who has joined us today. I think we peaked at almost 60 people turning up today, which is incredible to see. We've had some amazing questions from them as well. So thank you all for coming. I will once again remind people that this talk will be uploaded to YouTube if you would like to share it with someone, rewatch it, or if you have any follow-up questions. Again, the email address for Chloe you can see at the bottom of your screen currently. We do have more webinars happening next week, which you can find on the MASTS website or by following us on our Twitter. Any final closing remarks you'd like to say, Chloe?
Just thank you so much for this platform, and thanks for everyone for turning up. I'm chuffed at how many people have arrived, and yeah, I hope my answers to my questions were somewhat understandable. Been great. Cheers.
Thank you very much, and we'll hopefully see you all next week.